When I returned to Cambridge and broke the news of the DNA B form, Bragg no longer saw any reason for Crick and me to avoid DNA. He very much wanted the DNA structure to be found on his side of the Atlantic. So we went back to model building, looking for a way the known basic components of DNA, the backbone of the molecule and the 4 different bases, the backbone of the molecule and the 4 different bases, adenine, thymine, guanine, and cytosine, could fit together to make a helix. I commissioned the shop at the Cavendish to make us a set of 10 bases, but they couldn't produce them fast enough for me. I ended up cutting out rough approximations from stiff cardboard. By this time I realized the DNA density measurement evidence actually slightly favored a 2-chain rather than 3-chain model. So I decided to search out plausible double helices. As a biologist, I preferred the idea of a genetic molecule made of 2 rather than 3 components. After all, chromosomes, like cells, increase in number by duplicating, not triplicating. I knew that our previous model with the backbone on the inside and the bases hanging out was wrong. Chemical evidence from the University of Nottingham, which I had too long ignored, indicated that the bases must be hydrogen bonded to each other. They could only form bonds like this in the regular manner implied by the X-ray diffraction data if they were in the center of the molecule. But how could they come together in pairs? For 2 weeks I got nowhere, misled by an error in my nucleic acid chemistry textbook. Happily, on February 27th, Jerry Donahue, a theoretical chemist visiting the Cavendish from Caltech, pointed out that the textbook was wrong. So I changed the locations of the hydrogen atoms on my cardboard cutouts of the molecules. The next morning, February 28, 1953, the key features of the DNA model all fell into place. The 2 chains were held together by strong hydrogen bonds between adenine-thymine and guanine-cytosine base pairs. The inferences Crick had drawn the year before, based on Kargaff's research, had indeed been correct. Adenine does bond to thymine, and guanine does bond to cytosine, but not through flat surfaces to form molecular sandwiches. When Crick arrived, he took it all in rapidly and gave my base pairing schemes his blessing. He realized right away that it would result in the 2 strands of the double helix running in opposite directions. It was quite a moment. We felt sure that this was it. Anything that simple, that elegant, just had to be right. What got us most excited was the complementarity of the base sequences along the 2 chains. If you knew the sequence, the order of bases along one chain, you automatically knew the sequence along the other. It was immediately apparent that this must be how the genetic messages of genes are copied so exactly when chromosomes duplicate prior to cell division. The molecule would unzip to form 2 separate strands. Each separate strand then could serve as the template for the synthesis of a new strand, 1 double helix becoming 2. In What is Life, Schrödinger had suggested that the language of life might be like Morse code, a series of dots and dashes. He wasn't far off. The language of DNA is a linear series of A's, T's, G's, and C's. And just as transcribing a page out of a book can result in the odd typo, the rare mistake creeps in when all these A's, T's, G's, and C's are being copied along a chromosome. These errors are the mutations geneticists had talked about for almost 50 years. Change an I to an A, and Jim becomes jam in English. Change a T to a C, and ATG becomes ACG in DNA. The double helix made sense chemically, and it made sense biologically. Now there was no need to be concerned about Schrödinger's suggestion that new laws of physics might be necessary for an understanding of how the hereditary code script is duplicated. Genes, in fact, were no different from the rest of chemistry. Later that day, during lunch at the Eagle, the pub virtually adjacent to the Cavendish lab, Crick, ever the talker, could not help but tell everyone we had just found the secret of life. I myself, though no less electrified by the thought, would have waited until we had a pretty three-dimensional model to show off. Among the first to see our demonstration model was the chemist Alexander Todd. That the nature of the gene was so simple both surprised and pleased him. Later, however, he must have asked himself why his own lab, having established the general chemical structure of DNA chains, had not moved on to asking how the chains folded up in 3 dimensions. Instead, the essence of the molecule was left to be discovered by a 2-man team, a biologist and a physicist, neither of whom possessed a detailed command even of undergraduate chemistry. But paradoxically, this was, at least in part, the key to our success. Crick and I arrived at the double helix first precisely because most chemists at that time thought DNA too big a molecule to understand by chemical analysis. At the same time, the only 2 chemists with the vision to seek DNA's 3D structure made major tactical mistakes. Rosalind Franklin's was her resistance to model building. Linus Pauling's was a matter of simply neglecting to read the existing literature on DNA, particularly the data on its base composition published by Kargaff. Ironically, Pauling and Kargaff sailed across the Atlantic on the same ship following the Paris Biochemical Congress in 1952, but failed to hit it off. Pauling was long accustomed to being right, and he believed there was no chemical problem he could not work out from first principles by himself. Usually this confidence was not misplaced. During the Cold War, as a prominent critic of the American nuclear weapons development program, he was questioned by the FBI after giving a talk. How did he know how much plutonium there is in an atomic bomb? Pauling's response was, "Nobody told me. I figured it out." Over the next several months, Crick, and to a lesser extent, I, relished showing off our model to an endless stream of curious scientists. However, the Cambridge biochemists did not invite us to give a formal talk in the biochemistry building. They started to refer to it as the WC, punning our initials with those used in Britain for the toilet or water closet. That we had found the double helix without doing experiments irked them. The manuscript that we submitted to Nature in early April was published just over 3 weeks later, on April 25, 1953. Accompanying it were 2 longer papers by Franklin and Wilkins, both supporting the general correctness of our model. In June, I gave the first presentation of our model at the Cold Spring Harbor Symposium on Viruses. Max Delbruck saw to it that I was offered, at the last minute, an invitation to speak. To this intellectually high-powered meeting, I brought a 3-dimensional model built in the Cavendish, the adenine-thymine base pairs in red, and the guanine-cytosine pairs